Matangi of second year genetic engineering in SRM University. Today I'm going to present my topic about human genetic disorders. These are basically health conditions or disorders that are caused due to the abnormalities in the human gene. For example, in the gene structure, absence of a gene, presence of an additional gene, or in the chromosomal number. Types of genetical disorders. So my main focus of my presentation is on the categorization of genetic disorders. So there I have categorized them into 6 types: chromosomal aberrations, single gene inheritance disorders, multifactorial genetic inheritance disorders, mitochondrial genetic inheritance disorders, Mendelian inheritance patterns, and uniparental disomy. My first category is chromosomal aberrations. Chromosomal aberrations are referred to as the abnormalities in the chromosomal number or chromosomal structure. Chromosomal structural aberrations are caused due to mutations, which is 4 types: deletion, translocation, inversion, and addition. And change in the chromosomal number is of 2 types, which is euploidy and aneuploidy. Chromosomal aberrations, as I explained before, is of numerical and structural. Numerical, euploidy, aneuploidy. Euploidy is triploid, tetraploid, and polyploid. Aneuploidy is monosomy, trisomy, and heterosomy. Structural, deletion, duplication, inversion, and translocation. So some common examples of chromosomal aberrations include Klinefelter syndrome. So typically a human male contains 46 chromosomes with 2 sex chromosomes X and Y. But in this case, a human male contains an extra X chromosome, which is they contain 47 chromosomes X, X, Y. Down syndrome is a very common example of chromosomal aberrations. It is also known as trisomy 21. It is a genetical condition which is caused by the presence of an extra set of chromosome in the 21st chromosome. Single gene inheritance diseases. This is my second category. And these disorders are caused by mutations in a single gene. For some examples are cystic fibrosis, hemochromatosis, Tay-Sachs, sickle cell anemia. A very common example of single gene inheritance disorder is cystic fibrosis. Cystic fibrosis is an inherited disorder that is caused due to the mutations in a single gene called as CFTR, which is cystic fibrosis transmembrane conductance region. So mutations in that gene causes this disorder. This disorder is characterized by the excess secretion of mucus in the linings of organs, and the signs and symptoms include persistent cough, wheezing, exercise intolerance, lung infections, clogging of tubes that block free flow of air. My third category is multifactorial inheritance diseases. These disorders are caused due to several factors. One of the factors may be gene, but some other factors may include nutrition, lifestyle, addiction, medicines, illness, and pollution. Some examples of multifactorial inheritance disorders include birth defects, several types of cancers, blood pressure, diabetes, Alzheimer's, schizophrenia, bipolar, arthritis, skin conditions such as psoriasis, asthma, and allergies. So as you can see, genetic causes and environmental process. When both of these effects are combined, it can cause multifactorial inheritance disorders. So my fourth category is mitochondrial genetic inheritance disorders. So mitochondrial genetic diseases are affects how cells produce energy. They are caused by defects in the mitochondrial DNA encoded genes or nuclear DNAs whose products are imported into the mitochondria. These disorders can affect the mitochondrial mechanisms like mitochondrial DNA maintenance, pathogenic mtDNA variants, coenzyme Q10, respiratory chain complexes, mitochondrial structure. A common example of mitochondrial gene inheritance disorder is Leigh syndrome. Symptoms usually appear in infancy, but some cases may show in the early childhood or adulthood. So the common symptoms include loss of developmental milestones, seizures, abnormal body temperature, persistent vomiting, vision loss, difficulty in breathing, and heart disease. And my fifth category is Mendelian inheritance pattern-based disorders. So as we know, Mendelian inheritance patterns include 4 types, which is autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. So autosomal dominant, when a defective gene is present in the other chromosomes except the sex chromosomes, then it is called as dominant. And if the defective gene is dominant that in nature, so they have given here 3 examples: Huntington's disease, neurofibromatosis, and achondroplasia. Autosomal recessive includes if the defective gene is recessive, that means it should be in homozygous conditions to be expressed. So such disorders are called as autosomal recessive, cystic fibrosis, sickle cell anemia, Tay-Sachs disease. X-linked dominant. For example, if the defective gene is present in the sex chromosomes or allosomes, and if the defective gene is dominant, it can be in heterozygous condition or homozygous condition. Doesn't matter, but it will be expressed. So, the first example is vitamin D rickets, Fragile X syndrome, Alport syndrome. X-linked recessive. If the defective gene is if the defective gene is a sex chromosome, and it should be in homozygous condition to be expressed. Example: color blindness, hemophilia, muscular dystrophy, and Fabry disease. My sixth category is uniparental disomy. In the common cases, a growing fetus or a cells or the growing mass of cells will acquire one set of chromosomes from its mother and one set of chromosomes from its father. But in this case of uniparental disomy, the growing fetus only acquires one set of complete chromosome of either its mother or its father. So it can occur during the formation of egg or early in fetal development, and it acquires all the chromosomal pair from a single parent, and the other parent's chromosomes are missing. So examples of uniparental disomy includes Angelman syndrome and Prader-Willi syndrome. Thank you.